everyone. Thank you so much for coming, and thank you for all of you who might be listening in on Zoom. So I've given a couple. So I'm Valerie Paul. I'm the director of the Smithsonian Marine Station, working mostly over at the station, which is across the street from the aquarium, and also very happy to be involved with this wonderful facility here, the aquarium, the Smithsonian Marine Ecosystems Exhibit. So what I wanted to do today, I've given a couple of these talks over the years. A couple of them have been more about, overall research at the Smithsonian Marine Station and everything. But today I'm going to drill down to some of my own work and share a little bit with you about what I've been doing for actually multiple decades in some cases, but some recent work as well. So I'm going to go ahead and get started, and want to start off by acknowledging the team of people that has worked with me over the years, especially in recent years. None of this could be done by myself, and we have a tremendous team of people that have shared working with me on a lot of these things. I want to point out a couple people in particular who worked with me at the Smithsonian Marine Station for quite a number of years. First, Saraf Gunsakara, who is an extremely talented natural products chemist, and so he's been the mastermind behind a lot of the chemical isolation and structure determination of some of the compounds we've been working with. And Jennifer Sneed, who has worked with me for over a decade now, and she's a very excellent microbial chemical ecologist and does a lot of work with marine bacteria as well. And I'll talk a little bit about some of our work. And then in the back here, Jay Hope, who's also worked with us for about over a decade and has been a technician helping with many, many different projects. So really want to thank all of them for all their contributions to some of what I'm going to talk about today. So what I'd like to do in half an hour or so is quickly go through a lot of different things, to introduce you to marine chemical ecology, what that means, and talk a little bit about climate change and how that's influencing some of these changes that we see in our coastal waters and in our oceans. I'm going to talk a little bit about marine cyanobacteria. This has been a long time interest of mine for multiple decades now. And then more recent work on coral diseases and coral restoration activities. So before coming to the Smithsonian Marine Station, where I've been since 2002, I was at the University of Guam Marine Laboratory, where I went right after finishing my PhD research at Scripps Institution of Oceanography. This was an amazing experience. Guam is about halfway around the world from here, so keep going up to the middle of the Pacific, and if you go any further, you'll probably start heading back this way again. And our little marine station was down here on the water. This is the University of Guam up here on the top of the hill. And it was a great place to do field research and really get engaged with the tropical Pacific, learn a lot of the different organisms out there, and really start working pretty intensively on marine chemical ecology. So this is me driving a boat a long time ago, and some of the fairly simple tank setups and things that we had, but we had a great seawater system that piped the water in right off the reef, so it was a really great place to do all kinds of aquarium experiments and things. And then when I came to the Smithsonian Marine Station, I've been very fortunate to manage our little facility, our research facility, as well as since 2010, our little field station down at Belize, the Carrigo Key Field Station, where we also have excellent tropical research going on, and it's really a fun place to work. And so what have I been doing for all these years? Well, you're going to hear a little bit about that today, but I want to start by just introducing you to the idea of these natural products, these small molecules that are made by many different types of marine organisms, and what they do in the ocean. And I think many people are familiar with terrestrial plants you know, we know that some of them are toxic. They can make all kinds of deadly compounds. We also know about algal blooms and how those can be toxic. Many people know about red tide and ciguatera poisoning, and those are all made by marine algae and are toxic compounds. But many different marine organisms are producing these small molecules that can function as chemical defenses or various communication cues in terms of their natural functions. And so what you're seeing here are just these kind of chemical drawings, which these are all carbon molecules, just stick drawings. And then we show the other functional groups like oxygen and that sort of thing. And these small two molecules here are terpenes. And they're produced by these halimeda, these green algae, which if you've been snorkeling on the reef or if you look at our coral tank here, you see. And halimeda is a really cool plant, as well as some of these other green calcified green algae, because not only are they chemically defended, but they're also structurally defended. They're 80% or more by weight calcium carbonate, right? So you wouldn't think that would be very good fish food. Why would they also need these chemical defenses? But we've shown in various field experiments, I'll show you some pictures in a minute, that these will deter feeding by natural grazers. And so the halimeda, if you go out on almost any reef in the world, you'll see them there, and it's because they're very well protected. Not only do they have structural defenses, calcium carbonate, but also these chemical defenses as well. And so the way we have done these types of experiments in the field over the years is by making artificial fish food, like using palatable green algae and auger usually, and kind of like making like little jello cubes of artificial fish food. And we can embed these compounds into that to see if they're actually feeding deterrents or not, if they deter grazing. And in many cases they do. We've been able to demonstrate that sometimes fish will bite them and spit them out. Sometimes they go approach and don't even bite. And so we see all kinds of different activities in these kinds of field assays. And we can do these sorts of experiments in the aquarium as well. And so over the years, we've really been able to show that most of the algae that are very common on the reef in an area of high grazing activity with all these different fishes like parrotfish and surgeonfish that will eat, graze intensively on the reef, that the ones that are the algae that are abundant in these habitats are usually chemically defended. And so we know a lot about that now. We being, overall, like the coral reef ecology community, and we know that these compounds can play an important role in that. So, and here's just some examples of, fish grazing. This is a rabbit fish. We don't have these in the Caribbean, but they're common in the Pacific. Or, for instance, sea urchins, which also are important grazers, sea hares, which are a little bit of an exception because these guys can actually, can and will actually eat the more deterrent, chemically defended algae and use those for their own defenses. You know that they'll exude ink and other things, and so those can have some of the algal defenses in them. And in competitive interactions, I'll come back to this in a little while, but sponges are also very, very chemically rich and can use those compounds for their own defense as well. So over the years, we know that these small molecules can do all these different things. Everything from defense against grazers to competition for space. They can fight with each other chemically. They can protect themselves from being overgrown. That's anti-fouling activity. And of course, they can have antimicrobial activity. We know most of our antibiotics that we take are produced by bacteria. And so those can also play an important role in the marine environment as well. And then all these they can play various roles in spawning activity and other things. Seem to be stuck on my advance. Not quite sure why. Francis, do you have any sense of why it's not syncing the advance? So anyway, all of these examples, I won't go, obviously, I won't have time today to show you examples of all of these different things, but I would say that probably defense against grazers is one of the most important and probably most common things that we see. So all of this is happening, all these chemical interactions, chemical mediation of all sorts of activities, behavioral activities on the reef are happening in a changing ocean. So we know that CO2 concentrations are going up. That's contributing to warming seas, as well as a process called ocean acidification, where the pH of the seawater decreases, becomes more acidic slightly. And that is was subject of a science paper a number of years ago now that gave some different examples. This is from their examples from the Great Barrier Reef of what happens at different concentrations of CO2 and temperature increases. And the predictions were more simple and increasingly simple in less calcified habitats. We know it's more nuanced than this, but we can see examples of what happens with warming seas, and especially temperature, as I'll show you some pictures later, can play a really negative role on corals. So we've got these various changes taking place. The other change that is a little bit less well understood, and we're starting to work on a little bit more, is decreased oxygen in the seawater, or what can be termed hypoxia. And that also can occur because warming water holds less oxygen. So as some of these other changes take place, we also see water becoming more deoxygenated, and that can be also negative. Darn it.
Change.
There we go. And that just shows you some of the chemistry of these processes. And CO2 comes into our ocean water and dissolves in seawater. We get carbonic acid, and carbonic acid breaks down into bicarbonate and protons, and that's what drives the lower pH, this increasing level of protons in the seawater. What the consequences are is it makes it more difficult for calcifying organisms to form calcium carbonate and calcify. Going backwards. So with the Alameda in mind, we started doing some experiments at University of Miami, and here in our own facility that we have over across the street, these aquarium tank systems where we can independently control each aquarium for temperature, pH, and now oxygen as well. We've got them set up so that we can manipulate all three things. And this was work done by Justin Campbell, who was a postdoc at the time that he started working with me, and has since gone on to, he's a faculty member down at Florida International University in Miami. And this is Chris Langen, who's at University of Miami Rosenstiel School. And we were starting some of this work with him. So our subject is, again, this green algae Alameda. And in these aquarium systems, we grew them for over 2 months at 6 different levels of CO2. And we've also since done some CO2 temperature experiments. One of the interesting things was it took very high concentrations of CO2 to see any changes in their calcification levels. So they were a little bit not too susceptible to the CO2 changes that we were giving to them. But we were also measuring the levels of these compounds. Remember, I told you that they were involved in chemical defense of the plants, and those stayed fairly constant across all CO2 levels. So as we got to the higher CO2 levels, they were still producing these compounds. They're carbon-based compounds, so we wouldn't necessarily expect their levels to go down too much. But their calcification levels were a little bit less. So what we found was that they were a little less well protected against grazers because both the calcification and the compounds are playing a role in that defense against grazers. So I'm going to switch gears a little bit now and talk about a different kind of algae, cyanobacteria or blue-green algae. These are quite different from the green algae I just showed you, which are more complex organisms. They're eukaryotic. These are more like bacteria. They're very simple, in some cases single cell. What we're looking at here is a bloom of a very well-known problematic blue-green algae called Microcystis. It blooms in Lake Okeechobee. It has occasionally bloomed in our waterways. And this is a picture near the Roosevelt Bridge in Stewart of a very, very dense Microcystis or blue-green algae bloom a number of years ago. And this one was particularly bad. And what happens is this produces toxins called microcystins, and they are extremely toxic and can cause health problems, especially if ingested, but also just some evidence that they might be aerosolized, go up into the air a little bit as well, like red tide toxins do. And these are just a few more pictures. So unfortunately, we have been plagued by these over the years. There's a little bit less of a problem in recent years than some of the last big blooms were about 4 or 5 years ago. But they do pop up every spring and summer. They are responsive to warming water temperatures, and so we see these blooms every year to some extent or another. The other kind of cyanobacteria or blue-green algae, the one I just showed you is planktonic, so it's living up in the water column. It's little single cells, and the reason water was so green is that the density of those cells was so high that you could see, look like pea soup. We can also get blooms of these more of these bendic or bottom living cyanobacteria that look like a big mat of hair. Here in the Indian River Lagoon in the summer months, you'll see this kind of stuff, very common. Used to all be called Lingia. Sorry about that. And they just bloom up like, they can be in various colors. These are all different species, of course, different colors, different shapes. They can wash up on the beach, cause all kinds of problems. This sign on the bottom this morning is from Queensland, Australia. So it's a worldwide problem in tropical and subtropical waters. These are a little different than those single-celled ones because these are forming long filaments, microscopic view of it. And it almost looks like a coin roll where you have a roll of coins and all the coins are lined up like this. The cells are these little individual patterns here, and they line up to form kind of a pattern that looks like that. And then they're inside this polysaccharide sheath. So that's what every one of these filaments looks like microscopically. I mean, of course, the different species look a little different. The shapes of these cells varies, etc. But the taxonomy is pretty complex, and so most of that is done with molecular methods. So what causes these blooms? These are some pictures from Fort Lauderdale, Florida, down here on the bottom. When I first came to Florida from Guam in around 2004, 2005, we were having really dense blooms of these reddish, golden red cyanobacteria on the reef. Again, looked like hair, was overgrowing everything and really causing problems. Here's what the cells look like. And we know a lot of different things contribute. I mentioned warming temperatures, especially in the spring and summer months. These blooms can really take off. Light, of course, they're photosynthetic organisms, so they need light. Nutrient enrichment can really help fuel their blooms, and so there's a lot of focus in controlling blooms, especially some of those planktonic ones like Microcystis, on nutrient reduction. You hear a lot about that in the Indian River Lagoon. This is one reason why. Almost all sources of nutrient enrichment can contribute to these blooms. And they can have destructive effects. They can overgrow corals, reefs, seagrass beds, etc. So they are definitely nuisance organisms. However, from my perspective and those of some of my colleagues, we're really interested in the compounds they produce. These are really diverse compounds. Unlike the green algal compounds I showed you earlier, these contain nitrogen, so they often derived from amino acids. They're small, often cyclic, or most of these have some kind of cyclic molecule in them. And their diversity is really great. We keep finding more and more of these compounds. And one of the sources of funding for some of our work, I'm not going to talk a lot about this today, on the blue-green algae, has been the National Cancer Institute, because some of these compounds are very cytotoxic. And I've been working for many years with a colleague, it's kind of cut off up here on the top, named Lucie, who's at the University of Florida College of Pharmacy. And we've been working for many years with us collecting and discovering some of the compounds, working with him and his graduate students on that. And then he's very interested in more of the molecular mechanisms of how these compounds are working to be cytotoxic and to destroy cancer cells in some of their various assays. So these are just a few of the molecules that, in some cases, he's been working on for years. This was found in Guam, in Apra Harbor in Guam, and he's now worked out how it works, mechanism of action, and trying to further develop some of these compounds. So they're really interesting from that perspective, but it's kind of taking the approach of working with these toxic molecules to see how they could be potentially beneficial instead of just nuisance organisms. I showed you some of these ones that bloom in the seagrass bed. This is some work that Jenny C. did that we published years ago that showed that these compounds could be deterrent to sea urchins that live in the grass beds. And this is a simple little assay we developed years ago that works out really nicely. Again, it's this artificial fish food with green algae embedded in it to make it palatable. And then we can test some of the extracts and compounds. And the nice thing about it is we'll put the control food without any added extracts or compounds on one side, the treated food on the other, or vice versa. I don't know which one's here, but you can see that the grazers, when they're given a choice between these two foods, will completely leave this one alone and gobble this one up. So I'm presuming this was the control, but I don't actually know in this particular case. This is all red cyanobacteria. This is from our field station in Belize. And one of the people who have been doing, researchers who have been doing long-term monitoring in the Caribbean, such as down in Curacao, where they've had a field station, a marine station for many, many decades, are finding that one of the biggest changes to the reef is overgrowth by these cyanobacteria. And here's just a giant mat of this reddish stuff down in Belize on the reefs at around 40 feet. So we're seeing these kinds of changes occurring. Don't exactly know why, but think that both warming temperatures, and nutrient enrichment are playing a role. So what this means is that when you're getting a lot more overgrowth of cyanobacteria on the reef, you start seeing a lot more competition between corals here and cyanobacterial mats, like this gray one that's sort of surrounding this poor piece of coral. And then direct competition where one will start to overgrow the other. You can see a lot of examples of that here. This was that Fort Lauderdale bloom where the cyanobacteria was just growing over sea fans, and sponges, and everything. So some really interesting photos. And two that I'm going to talk a little bit about here in the next few minutes. Well, not too much about this one, but it's one of my favorites. It's called Caldora Salata. It's really interesting. I guess it's one of my favorites because it's laden with toxin. It's really, really interesting. Produced several of those that I showed you earlier, including one called Dolastatin 10 that has been researched for decades for anti-cancer properties. The other one that's really interesting, and this is actually a cyanobacterial mat, but what you're seeing a picture of here is something called black band disease. It's a coral disease that occurs around the world on over 60 species of corals. And we can see some really very prominent examples of black band disease in Caribbean reefs. This is probably a photo from Florida. And what happens is, this is live coral here. It's a nice golden color. It's got its symbiotic algae, endosymbiotic algae in its cells, and photosynthesizing. It's of course, coral is an animal, but it's also sort of a plant with the photosynthetic cells in it. This is dead coral over here that's starting to be overgrown by other algae, so it's completely dead. And then this white zone here is where it's been newly killed. So this is like really recent coral death. So it's pure white because you're seeing the skeleton of the coral. And then this black band disease is what's actually doing the killing of the coral. And so it moves this way, increasingly killing the live cells and causing mortality, potentially ultimately to this whole coral colony. I don't know, we didn't follow this one over time, but there are examples where it can kill a whole coral colony in weeks or months, depending on the circumstances. 2 summers ago, 2023, we had a really severe, some of you may remember, warming event in Florida's waters, especially down in the Florida Keys, but sort of from Miami southward. And these are some photos I took on the reef down in the Florida Keys. That I just wanted to show you. And what happens is in a coral bleaching event is they lose all those symbiotic algal cells and then just turn stark white. But in this case, these corals aren't completely dead yet. They're white because they've lost their algae, but the tissue is still there. And sometimes they come back if the bleaching event, and the bleaching is caused by, in most cases, warming water, but any severe temperature change can cause coral bleaching. We've seen coral bleaching in response to really cold water events as well. There was a hurricane that went through, it wasn't an immediate, I think it was Ian when it came through, caused a lot of, a real decrease in the temperature on some of the reefs in South Florida, and that also led to a bleaching event. But most often we associate it with warming temperatures. And then this example of this cyanobacterial mat completely overgrowing the seabed. And so this was a really, really extreme summer. We had lost a lot of corals in the Keys, especially the Acropora, the upright corals like you can see in our tank here. And just wanted to show you that example, but also show you this example on the next slide, where we were monitoring colonies off of Marathon, Florida. Throughout the summer, we were in the middle of a project on coral disease, which I'll show you more about in a minute. But I just wanted to show you this time series. These are Montestria cavernosa, star over coral, I think that's what it's called. But anyway, our project started in May and ended in December, so I don't have good pictures after December. But you can see how the coral went from nice golden brown, like we expect. I think the change here is just the lighting in the photograph. I don't think there's much change in terms of bleaching up until about July, and the real warm water event started about mid-July in 2023 down in the Keys. And you can see by August, this coral's lost virtually all of its pigment. The symbiotic algae are disappeared in that particular case. And it was really interesting to watch them recover. So here, August and September, they're both still pretty white, but you can see in October some little hints of recovery as the golden color starts to come back. This zooxanthellae returning to the colony for small amounts that were remaining starting to grow. We don't really know, regrow, but and you can see how it's often down, not on the very surface where they're really exposed to a lot of light, but kind of around the edges where the color is coming back. And then it just slowly starts to come back. You can see there's still a few portions of it that are bleached. Even in December, it hasn't fully returned, but it's mostly returned. And I thought that that was a really cool example of what happens over time when you get a major coral bleaching event. Fortunately, in this case, most of these Montesquieu recovered themselves. In fact, I think all of them recovered. So we were studying about 50 of them tagged that we were following. So I want to move into coral diseases, and the shift here is kind of a natural one if I talk about black band disease, because this is a cyanobacterial mat dominated by a particular cyanobacterium called Rhosiophilium, and Rhosiophilum, sorry, I'm saying it wrong. And so this one, I want to show you how we investigated some of these, some of the compounds that are being produced in here, and tried to understand what they're doing in the cyanobacterial mat. If the compounds themselves are toxins, they're playing any role in the disease process and the killing of the coral or not. And then I want to tell you a little bit about stony coral tissue loss disease. And this is a really, really bad disease that's been in Florida now for about a decade. So first, I'm going to quickly go through this black band disease story. I'm not going to take too much time on it, but as I mentioned, this Rhosiophilum is the dominant cyanobacterium. Here it is on a different species of coral, Orbicella babulata. It will, as I mentioned, attack almost any bolder coral. And the way we were collecting the mats, they're really challenging to collect because they're embedded down here into the skeleton. They're really well adhered and they're very thin, so it's not like there are tufts sticking up that you could really collect. But we use these syringes kind of suck off the mat. And this took a lot of time and a lot of work. We probably spent about 5 years on this project in terms of getting enough material to be able to study and really understand what was going on. This is a shot of Gonioptera from Guam. And the mat is a little bit harder to see here. It's kind of, this is sort of a finger coral, but it's kind of down here at the base of some of these polyps. So getting enough of that off to look at was kind of challenging. Here you see it on a brain coral. So it really can dominate a number of different species of corals. And these are the compounds we ultimately came up with. They're unique. They were unknown before we found them. It's as I mentioned at the Marine Station, took the lead on this project, but we worked closely with Hendrik's lab and also with microbiologist Julie Meyer, who's at the University of Florida, to characterize ultimately the biosynthetic gene cluster for these compounds and everything. So it was really a pretty cool study. But we call them the leukeolides because most of the black band disease that we got for this project we collected at Lucie Reef in the Florida Keys. And so what do these compounds do? Well, we found that they weren't particularly toxic. So the compounds themselves don't seem to be killing the coral. We couldn't get any effect from testing any organisms really with the compounds. They also weren't very strong antimicrobial compounds. So our other hypothesis was that they might be playing some role in signaling with other bacteria in the mats, because it's not just Rhosiophilum in the mats, it's all kinds of other organisms, other microorganisms, so bacteria and other cyanobacteria as well. But we couldn't really find a strong role for them as antimicrobial compounds either. So I have to ultimately confess that we still don't exactly know what they're doing, but we do know that they play a role in reducing reactive oxygen species. And you all know that eating antioxidants, like things that have good like color to them, is good for you because they have pigments in them that are antioxidant molecules. And so we think that these compounds might be helping the producing organism themselves in the process of overgrowing the coral because corals themselves have their own defenses. One of the first things they do is produce reactive oxygen species when they're being attacked, and such as hydrogen peroxide. And so we were able to show that. Ultimately, that's as far as we got on this particular project. And the reason why we kind of published what we had and didn't work on it too much more after that, or haven't been working on it very much, is because of this much more immediate and devastating disease called stony coral tissue loss disease that popped up in Florida right in 2014, right around Miami, and then quickly spread over coming years up and down the entire Florida reef tract, and ultimately got to the dry tortugas in 2021. So now it's everywhere in Florida. You don't see it as much as we did. Many hundreds of thousands of corals succumb to this disease, and unfortunately, it's now spread throughout the Caribbean. And the only part of the Caribbean, so recently, last year, I got reports that it had popped up in Costa Rica. So the only part of the Caribbean where we don't find it now is Panama. And so the Smithsonian conveniently has field stations down in Panama, Smithsonian Tropical Research Institute. We're hoping to get down there later this year and get some good baseline data before, unfortunately, we feel inevitably that it will show up there. And then there is some concern about whether it could make its way across the Panama Canal into the Pacific, but we don't know yet very much about how susceptible some of the Pacific corals might be to this disease. Just an example of how rapidly it can kill a coral. This is some photos sent to me by colleagues at the U.S. Virgin Islands. A big brain coral. And about 6 weeks from when the disease first starts showing up as these little white patches, that's just tissue loss, death of the colony. To increasingly, ultimately killing the colony and just leaving this white skeleton behind. So we've been doing experiments since 2017. We were able to get some National Science Foundation rapid funding to start working on this. And I started working with a collaborator, Greta Abbey, who's was at University of Hawaii, and she is a coral disease expert. She's worked on coral diseases around the world, and she's continued to stay interested in studying coral tissue loss disease. So we set up our little experiments out on our outside slab, wet lab area, so that they could get natural lighting, and set up a lot of individual tanks. So each of these might hold a piece of coral. We were able to do a lot of different experiments, everything from treating with antibiotics to show that could slow or halt the disease. We still are searching for, as are other research groups, the causative organism. Nobody knows what is causing this disease to this day. It's been a decade now. But one of the things we started looking at, because we knew that antibiotics could slow or halt the disease, they've since used those a bit on the reef itself to treat the corals. We started looking for probiotics or beneficial bacteria to see if we could find something that might be a treatment, or at least give us some insights into stony coral tissue loss disease. And probiotics are, of course, used in humans, of course, as well as many different aquaculture situations. And these are just some examples of products that are available already. And so we have a library of about 2,000 or more coral isolates. We started screening them for antimicrobial activity to see if we could find one that might have some effect against whatever kind of causative organisms we have in stony coral tissue loss disease. Of course, we are still working a bit blind because we don't know what causes stony coral tissue loss disease. But we had a big kind of a procedure where we would grow up bacteria, grow them up in larger quantities, screen for antimicrobial activity. And one of the first ones we were working with actually ended up being kind of a lucky first grab. We got one, this golden colored bacterium here growing on just an auger plate. And you can see around it is this kind of clear zone. And what we do is we streak the plate with the lawn of our test bacterium, put the other bacterium on the plate, and look for these zones where the test bacterium doesn't grow, right? So showing that it's got some antimicrobial activity. We were also ultimately able to isolate a few compounds from it, chloromycin, which is a known compound from first isolated in Koror in Palau. So it's clearly present in these bacteria, Pseudoultramonas species around the world. And then we knew it produced these enzymes that can create reactive oxygen species to kill other bacteria. And then it also produces this small molecule I'll come back to very quickly at the end, called tetrabromopyrrole. So this thing, when we tested it in aquarium assays, could slow or stop the disease in some cases. We published this last year. And then we tried to take this out into the field. So how do you take something that works in a little aquarium and test it in the field? Well, we developed this sort of plastic bagging method, and I'm going to credit Kelly Pitts, who worked with us for leading a lot of this work, and then injecting the probiotic into the bag. We got, of course, all our permits to test this. And we're now trying to publish a paper that shows that this more or less worked. We also developed this paste method that didn't seem to work quite as well. And then tracked the corals that were treated in this first case for almost 3 years with 3D models to determine the growth, how if we could slow or stop the progression of this disease. So we had to measure the amount of live tissue on the colony over time and compare it. And these models were super helpful. So I just want to show you at the end of the 3-year experiment how much tissue remained. And our probiotic bagging method seemed to do the best. We had about 95% coral tissue remaining, whereas our control for the bagging method was only around 60%. Our background controls were even lower. And the paste, surprisingly, with the probiotic incorporated, didn't do very well at all. We're not exactly sure why, but we sort of dropped that method after that and went with the bagging method. So we've stopped doing the testing for now. We have some good results from both. That was work done at both Fort Lauderdale and Broward County, but also down in the Keys, we have some testing that we did where it didn't work as well. We aren't entirely sure why, but we know that the disease in the Keys is a little bit different than it is up further north on the reef tract. It seems to be more virulent and cause coral death quicker. So not 100% sure why that is, but it is an interesting finding. So we've kind of wrapped up the testing. We don't see as much disease on the reefs now, 10 years later, as we did. So I think people have stopped treating for the disease in general. And then I finally want to just wrap up real quickly in the next 5 minutes, talking to you a little bit about some coral restoration work. And really the motivation here is how do you restore a reef that's degraded with low coral cover, which is unfortunately most of our reefs in Florida now, to something that looks a little bit healthier with more live coral cover, less algae, etc. And there is a lot of work going on with coral restoration throughout Florida, the Florida Keys, in South Florida, in Miami, and Broward County. And we've been partners with a lot of these folks for the last 5 or 6 years. First with some funding from DARPA, Defense Research Program, and now with some NOAA funding to try to develop some more advanced methods and use some of these tools like probiotics and settlement cues to further advance reef restoration rather than just simply cutting coral and letting it regrow, but to try to move things along faster and use some additional tools. So this just takes you through a coral life cycle. This is Brydes. It's a brooding coral. It releases larvae right after the new moon, and usually in Florida it's in April and May. And here you can see a larva and a newly settled coral polyp. So there's several of them here in this photo. And then they ultimately grow and form new coral. So a lot of our work has been done in Caribou Key, mostly because we have such great coral stands right next to the island. There's you can't see it in this photo, but it's our tiny little island, our field station. But right around this side of the island is a really great Crocodile palmata stand, the big elkhorn coral. And here you can see a spawn at night. It's an amazing site in a dense spawn because all of these little pink bundles, pink blobs that are up here in the water column are bundles of sperm and egg of Crocodile palmata, elkhorn coral. So it's almost like diving in a snow globe when you're out there at night. It's really, really impressive. So we can catch the spawn by putting these nets over them. Here it floats because the eggs are so fatty that that sperm egg bundles float up. Here we caught them in this plastic jar that we put at the top of the end of the net. And then we can bring them back to the lab, simple little laboratory setup with a good seawater system down in Caribou and rear them up. And in about 4 or 5 days, you have larvae. And we've been able to do different experiments where we can put the larvae in these chambers and give them different kinds of settlement cues, things we know they like coral and algae, things we know they don't like macroalgae, and see how they settle and grow. And so here's just one example where this blue-green algae, cyanobacterium called Homothamian, significantly inhibited settlement when we did those kinds of studies. And we can track some of that to the fact that they release the compounds into the seawater as well. We used really good offshore water. This is work done by a former postdoc, Skylar Carlson, and just bubbled the Homothamian in some seawater, put it over columns to the resin columns. This is carbon C18 resin, and then that tracks the compounds. And by mass spec, we could show that we could pull this particular compound, laxophysin A, its mass. Here's the peak itself in a chromatogram, and here you can see it matches up to with the whole organism. So we can see that compound's in there. So it works out pretty well. And let's quickly go through. So we know that some things like blue-green algae can inhibit settlement. We also know that some things like crustose coralline algae, this little pink it's a red alga that encrusts corals and rocks and everything, good, healthy, well-raised environment in the tropics. And we were doing choice experiments to see if coral larvae pick one species of CCA over another. These aren't that easy to tell apart. You have to use microscopic methods and even molecular methods to really do it well. But I like this next photo because it shows you really well. This is also a paper we published where we ultimately figured out the preferences for 7 different Caribbean corals. This was all work done, mostly almost all work done in Caribou. This is what the larvae look like when they're starting to explore the coral and algae or any substrate to find kind of where they want to settle. And settlement for an organism that stays attached to the bottom is a really important step because this is a way of picking a suitable habitat for their entire rest of their life, their adult life. And then they undergo metamorphosis. I think everybody can think of metamorphosis in terms of a caterpillar to butterfly, right? That's metamorphosis. It's the same organism, but it undergoes a very relatively quick and dramatic change in the way that the life history and the way the organism looks. So this is a coral larva that is adapted to swim around, well, not swim all that well, but to float around in the water column, do a little bit of swimming down to the bottom and explore the surfaces. And then this is a newly settled coral polyp of the same species. This is a crocodile. So beautiful kind of pebble-shaped pattern, and then it's kind of hard to believe that this one little coral will grow into a whole new elkhorn coral. This is me putting some settlement tiles down. We've settled them out on different substrates, put it back on the reef, and see how well the larvae survived. Well, some new settlers survived, not all that well, I should say, but a few of them do survive and start to grow. And this process of sexual reproduction is being incorporated more and more into reef restoration as well, because that way the corals, there's some more genetic variability than just cutting and letting them regrow, which is just cloning basically. So I just want to point out one molecule, this tetrabromoferol that I talked about earlier. Jenny across the street has been really involved in using it as a settlement cue, and we recently published a paper earlier this year or early last year that showed that by working with a colleague who synthesized a bunch of very similar molecules to tetrabromoferol, that corals would only settle and metamorphose in response to this compound. Nothing else worked. We were trying to look for other molecules that might be either simpler to make or a little more stable, because tetrabromoferol is not a very stable molecule. It degrades pretty easily. But the signaling process is very specific, and this compound is a really good settlement cue for every coral species that we work with in the Caribbean. So it's pretty cool. So what are all the implications of why is chemical ecology even important in reef restoration? Well, certainly we know that the cues are important, right? I just showed you the TVP cue, how specific they can be. Really, we only have certain molecules that even work, and we're in the process of pulling other molecules out that work. We found that Bill's deep refuge tank is a really good source of excellent chemical cues for settlement, but we don't know what's in there that's important. But so he always wonders why we keep coming over to dip more water out of this tank. That's why. So really, these cues are so important for the reproductive process. Settlement cues and we know that many different organisms, invertebrates, fishes, etc., perceiving cues for settlement. It's kind of like a big giant unknown about what some of those molecules are. We know that certain CCA are important, but now we've learned that even waterborne cues that are being released from the CCA are important. So there's a really lot of interesting chemical ecology remaining in the whole coral reproduction process. And so we're very interested and eager in figuring out what maybe some of these molecules are and applying them more and more to reef restoration. So I just want to thank a lot of people. This is my colleague Kendrick that I partner with at University of Florida on all of our exploration of molecules for biomedical purposes, but all of our various postdocs and members of the team over at the Marine Station. I mentioned Greta and all the folks that work with us on all the coral disease research, and just a great team of people and collaborators and everything that's helped make this possible, as well as funding, which is important, of course, for supporting a lot of these folks and getting the work done. So I'm happy to take a question or two. It's getting to be 10 o'clock already, but if anybody has any questions, I can address them now.
Did you find any sort of relationship between the morphology of like how the cyanobacteria colony forms and how much or what kind of toxins they have?
I don't know if everyone can hear very well, but the question was, is there any relationship between the morphology of the cyanobacteria and the toxins? And the short answer is yes, and that's because the morphology of these cyanobacteria is very much related to their species identity. And we know that different species of cyanobacteria are producing different compounds, but it actually took a long time for people to, it seems like the most simple and obvious answer, but the problem is that you can't really tell these cyanobacteria apart all that well. And if you look at them under the microscope, they kind of all look very similar. And so it wasn't until more and more molecular tools were being applied to the taxonomy and systematics of cyanobacteria that we were beginning to understand a little bit more about what the species identity even was and what was related to what. So now more and more we're applying with collaborators whole genome methods to being able to pick out. I mean, because they're small genomes. They're not like humans or anything. They're bacterial genomes. A little bit bigger than some bacteria because a lot of the genome is associated with these toxin-producing genes. I mean, they're just amazingly diverse. And cyanobacteria, have been on the planet for 3 billion years, so they've figured it out. But we're learning more and more about them all the time.
Yes, maybe I missed something. Do you have any hypotheses on why that red encrusted algae would produce a chemical that promotes settling?
Yeah, I mean, I think that's a good question. So the question is, why would crustose coral and algae, which I've heard people call them, they look like pink paint on rocks. They really are thin crusts overgrowing much of the reef. And we've known for a long time that a healthy reef is dominated by corals and a lot of coral and algae. And so why would they produce a hue that corals would perceive? And I think it's more that the corals have adapted to somehow to perceiving these hues as a sign, as an indication of a good place to settle. I mean, how that evolution, how that's happened evolutionarily, I don't really know. And it also doesn't help that we don't really have, besides TBP, which actually TBP is produced by bacteria that live on the coral and algae, not by the coral and algae itself. So in some cases, it's the biofilm, the bacteria that are living on these surfaces that can induce the settlement. In other cases, we think the coral and algae itself, coral and algae themselves are producing the compounds, but it's really, there's a lot of unknowns. The bacteria are easy to work with. I mean, that's how we first found, Jenny was working, I think when she was still a postdoctoral researcher on this, how we first found that the bacteria were producing them. We were isolating different bacteria from surfaces and found that these particular bacteria induced coral settlement, and then traced it down to this little molecule. But yeah, in that case, it was the bacteria that's doing it. In other cases, because we can treat the bacteria, the coral and algae with antibiotics. You can't completely get rid of all the bacteria by treating with antibiotics, but you can knock it way back, and they're still inductive. So we think that there's also stuff in the coral and algae itself. And there was a team of researchers in the 1980s now, Dan and Eileen Morse at UC Santa Barbara, who were some of the first to show that you could pull molecules from coral and algae that could induce coral settlement. So people have known that for a long time, but there's just still work ongoing on what those compounds are.
And they don't compete for substrate, or?
They do. They'll even overgrow each other. Different species of coral and algae will overgrow each other. Yeah, they're good in a good reef where they're not being outcompeted by turf algae or macroalgae. You'll see a lot of coral and algal dominance, and they aren't particularly easy to tell apart either. In terms of putting a taxonomic name on them, we've learned to pick out a few that we know are good ones for settlement, but the diversity is much higher than we fully appreciate. I mean, there are of course experts that are working on these, but our field IDs are limited to a few species that we feel confident in being able to ID. Well, thank you all so much. Appreciate it. Thank you to those who are online watching, and I think this is being recorded, so we have sample that we can put up at some later date. So thank you all.
Thank you.